welcome to part 2 of the NCERT reading of the sexual reproduction of flowering plant. We will continue from pollination. In the preceding sections, you have learned that the male and female gametes in flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into three types. The first one is autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. Figure 1.9a. In normal flower, which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, that is common pansy, oxalis and commelina produce two types of flowers. Xenogamous flowers, which are similar to flowers of other species with exposed anthers and stigma, and cleistogamous flowers, which do not open at all. Figure 1.9c. In such flowers, the anthers and stigma lie close to each other. When anthers dehiscence in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous as there is no chance of cross-pollen landing on the stigma. Cleistogamous flowers produce assured seed set even in the absence of pollinators. Do you think that cleistogamy is advantageous or disadvantageous to the plant? Why? The second one is Geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although Geitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. Xenogamy. Transfer of pollen grains from anther to the stigma of a different plant. Figure 1.9b. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agents of pollination. Plants use two abiotic, wind and water, and one biotic, animals, agents to achieve pollination. Majority of plants use biotic agents for pollination. Only a small portion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that the pollens are easily dispersed into wind currents. Figure 1.10. And large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The ears you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower plant groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of water for transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zostera. Not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of water by the long stalk, and the male flowers of pollen grains are released onto the surface of water. They are carried passively by water currents, figure 1.11a. Some of them eventually reach the female flowers and the stigma. In other group of water pollinated plants such as sea grasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long ribbon-like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Majority of flowering plants use a range of animals as pollinating agents: bees, butterflies, flies, beetles, wasps, ants, moths, birds like sunbirds and hummingbirds, and bats are the common pollinating agents. Figure 1.11b. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals, such as some primates, that is lemurs, arboreal, that is tree dwelling rodents, or even reptiles like gecko lizard and garden lizards, have also been reported as pollinators in some species. Often, flowers of animal pollinated plants are specifically adapted for a particular species of animal. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flowers pollinated by flies and beetles secrete fall odors to attract these animals. To sustain animal visits, the flowers have to provide rewards to these animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitors comes in contact with the anthers and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe place to lay eggs. An example is that of the tallest flower of Amorphophallus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant yucca, where both species, moth and the plant, cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary and the flower, in turn, gets pollinated by the moth. The larvae of the moth come out of the eggs as the seeds start developing. Why don't you observe some flowers of the following plants or any others available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animals visit them and whether they could be pollinators. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visits it. Carefully observe if any of the visitors came in contact with the anthers and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred to as pollen or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Breeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower or the other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not Geitonogamy. In several species such as papaya, male and female flowers are present on different plants, that is, each plant is either male or female dioici. This condition prevents both autogamy and Geitonogamy. Pollen-pistil interaction. Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from the other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type, that is compatible, or of the wrong type, that is incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grains germinates on the stigma to produce a pollen tube through one of the germ pores, figure 1.12a. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary, figure 1.12b and c. If you would recall that in some plants, pollen grains are said at two-celled conditions, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Pollen tube after reaching the ovary enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus, figure 1.12d and E. Many recent studies have shown that filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. All these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen-pistil interaction. As pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction, even in incompatible pollinations to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam, and vinca on the glass side, containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma, and the flowers are rebagged and the fruits allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the Xenogamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located at the central cell and fuses with them to produce a triploid primary endosperm nucleus, that is PEN, figure 1.13a. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two types of fusions, Xenogamy and triple fusions, take place in an embryo sac, the phenomena is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, that is PEC, and develops into the endosperm while the zygote develops into an embryo. 1.4 Post-fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruits are collectively termed post-fertilization events. Endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the span undergoes successive nuclear divisions to get rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans, seed maturation, or it may persist in the mature seed, example castor and coconut, and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledonous embryo. The zygote gives rise to the pro-embryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo, figure 1.14a, consists of an embryonal axis and the two cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle, that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons, figure 1.14b, possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of the embryo and the seed. Seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seed is formed inside fruit. A seed typically consists of seed coats, cotyledons, and an embryo axis. The cotyledons, figure 1.15a of the embryo, are simple structures generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example pea, groundnut. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. Example wheat, maize, barley, castor. Occasionally in some seeds such as black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucleus is the perisperm. Integuments of ovules harden as tough protective seed coats. Figure 1.15a. The micropyle remains as the small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seed becomes relatively dry, that is 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter the state of inactivity called dormancy, or if favorable conditions are available, that is adequate moisture, oxygen and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into a fruit. That is, the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have evolved mechanism for dispersal of seed. Recall the classification of fruits and their dispersal mechanism that you have studied in the earlier classes. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits, figure 1.15b. Most fruits, however, develop only from the ovary and are called true fruits, although in most of the species fruits are the result of fertilization. There are few species in which fruits developed without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and help the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variations. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds which can be used as food throughout the year and also to raise crop in the next season. You can imagine agriculture in the absence of seeds, or in presence of seeds which germinates straight away soon after formation and cannot be stored. How long do the seed remains alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within a few months. Seeds of the large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of lupine, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of a date palm, that is Phoenix dactylifera, discovered during the archaeological excavation of King Herod's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions: how many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruits of some parasitic species such as Orobanche and Striga. Have you ever seen a tiny seed of ficus? How large is the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? 1.5 Apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants, such as some species of asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction, division, and develops into the embryo without fertilization. More often, as in many citrus and varieties, some of the nucellar cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different size and shapes from each seeds. Count the number of embryos in each seeds. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrid is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, and hence the cost of hybrid seeds become too expensive for the farmers. If these hybrids are made into apomictics, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year because of the importance of apomixis in hybrid seed industry. Active research is going on in many laboratories around the world to understand the genetic of apomixis and to transfer apomictic genes into hybrid varieties. Summary: flowers are the set of sexual reproduction in angiosperms. In the flower, androecium consisting of stamens represent the male reproductive organs, and gynoecium consisting of pistils represent the female reproductive organs. A typical anther is bilobed, thecus, and tetrasporangiate. Pollen grains develop inside the microsporangia, four wall layers, the epidermis, endothecium, middle layers, and the tapetum surrounded the microsporangium. Cells of the sporogenous tissue lying in the center of the microsporangium undergoes meiosis, that is microsporogenesis, to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represents the male gametophytic generation. The pollen grains have a two-layered wall, the outer exine and inner intine. The exine is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and generative cells, or three cells, a vegetative cell and two male gametes at the time of shedding. The pistil has three parts: the stigma, style, and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus, in which the archisporium differentiates. A cell of the archisporium, the megaspore mother cell, divides meiotically, and one of the megaspores forms the embryo sac. The female gametophyte, the mature embryo sac, is 7-celled, 8-nucleated at the micropylar end. Is the egg apparatus consisting of two synergids and an egg cell. At the chalazal end are three antipodals. At the center is a large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic, wind and water, or biotic, that is animals. Pollen-pistil interaction involves all events from the leading of landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style, enter the ovules and finally discharges two male gametes in one of the synergids. Angiosperms exhibit double fertilization because of two, two fusion events occur in each embryo sac, namely Xenogamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develops into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the pro-embryo, globular and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of seeds without fertilization. Apomictics have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony. And that's our chapter is finished. Thank you for watching and stay tuned for the NCERT reading of the next chapter. If this video helped you, then like, share and subscribe.